You blink your eyes, and it takes about 350 milliseconds. In that exact same window, a star-nosed mole could have touched, evaluated, and made eating decisions about 44 separate objects. And it could have swallowed one of them before your eyelid even reopened. This blind, hamster-sized animal living in total darkness under the swamps of eastern North America holds the Guinness World Record for the fastest eating mammal alive. Its brain decides whether something is food in 8 milliseconds, which sits right at the physical speed limit of how fast neurons can fire. No nervous system on the planet operates faster. And the tool that makes all of this possible, 22 pink fleshy tentacles exploding out of its face, packed with 100,000 nerve fibers crammed into an area smaller than your fingertip. The creature that looks like evolution went horribly wrong is actually one of the most neurologically optimized animals science has ever studied. In the early 1990s, a young undergraduate named Kenneth Catania was working as a research assistant at the National Zoo in Washington, D.C., collecting small mammals from swamps and marshes in Pennsylvania. One of those mammals was the star-nosed mole, and the moment Catania examined its brain, he realized something extraordinary was hiding behind that nightmare face. He built his entire career around it. 3 decades at Vanderbilt University, picking apart how this animal works. In 2006, the MacArthur Foundation handed him a $500,000 Genius Grant with no strings attached, largely because of what the mole's nose taught the world about how brains process sensory information. He went on to win the Pridal Research Award from the National Academy of Sciences in 2013, and published a book called Great Adaptations through Princeton University Press in 2020, starring the star-nosed mole as its centerpiece. Catania himself put it best. He called the star-nosed mole among the weirdest-looking creatures on the planet, but then added, "When I began trying to understand the star, the mole's brain organization, and its behavior, that's when things got really surprising." And the surprises started with the nose. What Catania found over the next 3 decades would challenge everything scientists assumed about how mammalian brains are built. Those 22 fleshy appendages fanning out around the nostrils are called rays. 11 on each side, constantly in motion, sweeping the environment like tiny pink fingers. Each ray is covered in thousands of microscopic raised domes, roughly 30 to 50 micrometers across. These are Eimer's organs, first described in 1871 by German zoologist Theodor Eimer when he studied the European mole. Every mole species has Eimer's organs, but the star-nosed mole packs about 25,000 of them onto a surface barely 1 centimeter across. 6 times more touch sensors than the entire human hand, squeezed onto a nose smaller than a fingertip. Kenneth Catania described the star's skin as so sensitive that his team was never able to find the lowest threshold needed to trigger a neural response. They kept reducing the stimulus, and the mole kept detecting it. But what Catania discovered about how the mole actually uses these sensors is what blew the whole field open. When a star-nosed mole moves through its tunnel, the star touches 10 to 13 spots every single second, scanning the walls and ground in rapid, sweeping patterns. The moment one of the outer rays contacts something interesting, the mole snaps its head so that one specific pair of rays, the 11th pair at the bottom of the star, lands directly on the object. That movement takes about 50 milliseconds, and it looks exactly like a saccade. Those fast, jerky movements your eyes make when you shift your gaze from one thing to another. Catania named this the somatosensory fovea. In your eye, the fovea is a tiny spot in the center of the retina that gives you your sharpest vision. Your eyes constantly dart around, so whatever you want to examine in detail lands right on that fovea. The star-nosed mole does the exact same thing, but with touch instead of light. The 11th ray is the high-definition center, and every movement of the star repositions it onto objects that matter, and the brain confirmed it. Over 52% of the mole's somatosensory cortex, more than half the brain's touch processing territory, is devoted to the nose alone. The nose makes up only about 10% of the animal's body, but the brain assigned it the majority of its processing power. The 11th ray, despite being the smallest on the star, takes up 25% of the primary somatosensory cortex representation all by itself. Evolution built an eye out of touch receptors and then wired a brain to match. In 2005, Catania published a study in the journal Nature using high-speed cameras to film star-nosed moles foraging. He measured what scientists call handling time, the total span from first touching a prey item to eating it and moving on. The average came out to 227 milliseconds. The fastest? 120 milliseconds. That is more than twice as fast as a human blink. It takes a human driver roughly 650 milliseconds to hit the brakes at a red light. This mole finds, identifies, captures, and swallows a worm segment before your foot even reaches the pedal. Guinness certified it as the fastest eating mammal on record. And in the lab, Catania filmed moles locating and consuming 8 separate prey items in under 2 seconds, then immediately resuming the search without pausing. And the decision speed behind that number is where things get truly wild. From the instant the star touches something, neurons in the somatosensory cortex respond within 12 milliseconds. Motor commands travel back to the star in another 5 milliseconds. The full decision cycle, edible or not, wraps up in about 8 milliseconds total. That same process takes 600 milliseconds in a human. The mole's nervous system is running at the physiological ceiling of how fast neurons can conduct signals. Catania's data showed the moles sometimes lunge in the wrong direction before correcting, which told him they are literally operating at the limits of their own brain's processing speed. Why push that hard? Because each individual bug or worm in a wetland marsh provides almost no calories. If the mole spent even half a second evaluating each one, it would burn more energy hunting than it recovered from eating. So evolution compressed handling time as close to zero as biologically possible. Catania's analysis placed the star-nosed mole right on the theoretical asymptote of foraging profitability. There is almost no room left for improvement. This animal hit the ceiling. And then, Catania caught them doing something the entire scientific community had declared impossible. While filming moles in a glass-bottomed aquarium, he noticed they kept blowing tiny air bubbles out of their nostrils and immediately sucking them back in. The rhythm was about 5 to 10 bubbles per second, almost identical to the sniffing rate of rats and mice on dry land. That caught his attention immediately. He set up a test. Underwater scent trails leading to food were laid on 2 random paths inside a tank. A steel grid covered the trails, fine enough for bubbles to pass through, but too tight for the mole's tentacles to touch anything below it. 5 moles tested followed the correct scent path between 75 and 100% of the time. When Catania switched to a finer grid that blocked the bubbles too, accuracy crashed to 50%, pure chance. The moles were blowing bubbles onto objects and scent trails, picking up dissolved odor molecules on contact, and inhaling them back to wash those molecules across the olfactory receptors inside the nose. Each bubble measured between 0.06 and 0.1 millimeters. He published the results in Nature in 2006. Before that paper, the scientific consensus held that it was flatly impossible for any mammal to smell underwater. Catania proved them wrong with a mole and a fish tank. The star-nosed mole and the American water shrew became the first 2 mammals ever caught using olfaction beneath the surface. Since then, researchers found the same ability in the Russian demand too. One weird creature, as Catania put it, opened the door to an ability that turned out to be much more widespread than anyone had imagined. So what kind of body houses all of this? The answer is surprisingly unimpressive to look at. The star-nosed mole weighs about 40 to 55 grams, stretches 15 to 20 centimeters long, and lives just 2 to 4 years. It is functionally blind, with tiny eyes that pick up little beyond light and dark. Thick blackish-brown fur repels water completely. The tail swells up to 3 or 4 times its normal size in winter, storing fat for the cold months. And those oversized front claws dig tunnels that stretch over 100 feet long, sometimes opening directly into ponds and streams underwater. This is the only semi-aquatic mole species in the world, and it runs on a metabolism so intense that it eats roughly half its body weight every single day. Active day and night, year-round, even foraging under ice in winter. Worms, aquatic insects, crustaceans, mollusks, larvae, small fish, small amphibians. Anything the star confirms as food in 8 milliseconds goes straight down. They mate in late winter, give birth to litters of 2 to 7 pups in late spring. Pups arrive hairless, with ears, eyes, and stars completely sealed shut. Everything opens around 14 days later, full maturity at 10 months. Hawks, owls, foxes, minks, weasels, large fish, and even bullfrogs hunt them, but the mole's ability to vanish underwater or disappear into a tunnel in seconds gives it an escape edge that most small mammals never get. What makes this animal impossible to walk away from is the design principle hiding behind that face. Your visual system built a small, high-resolution fovea surrounded by lower-resolution periphery. Your eyes dart around in saccadic movements to land that fovea on whatever you need to examine. Bats built the same thing with sound, creating an auditory fovea for processing echolocation frequencies. And the star-nosed mole built the same architecture with touch. A small 11th ray fovea surrounded by broader scanning rays with rapid saccade-like movements dozens of times per second. 3 completely different senses, one identical solution. As Catania described it, evolution has repeatedly arrived at the same answer for building a high-resolution sensory system. Subdivide the surface into a large low-resolution area for scanning, and a small high-resolution area for focusing on what matters. The animal that looks like evolution's worst mistake is actually running the same sensory blueprint as your own eyes, just built out of touch receptors instead of photoreceptors. 25,000 Eimer's organs, 100,000 nerve fibers, a brain operating at the speed limit of neural conduction, a Guinness World Record for eating speed, and the first proof that mammals can smell underwater. All packed inside a face that would give you nightmares. After 3 decades of research, Catania says the star-nosed mole still has surprises left to reveal. As he told an interviewer, "I use the word amazing a lot to describe the star-nosed mole, and for good reason. It is a true neurological wonder that has taught us a lot about how brains process sensory information." And scientists still do not fully understand how Eimer's organs convert physical touch into the electrical signals that those 100,000 nerve fibers carry to the brain. The mechanism that makes the whole system work remains, after 150 years of study, an open question. 30 years of research, and this face still has secrets. If you want to see what other creature turned out to be an evolutionary masterpiece hiding in plain sight, hit subscribe and tap the video on screen right now.